Friday, October 11th. As usual, we're going to get started with a brief introduction here, and then I'll introduce and hand control over to our speaker. Although before we begin, I just wanted to open the floor in case anyone has any announcements that they would like to make. You can feel free to unmute yourself and make the announcement if you'd like, or you can email us and we'll make the announcement at the next session. All right, then just a reminder that after today, we have 4 more speakers here in fall 2024. It's a nice lineup going all the way through December 6th. We'll pause briefly over winter break and we'll resume on January 10th. And as usual, you're always welcome to nominate future speakers, including yourself, if you'd like. You can find the link on the webpage in the top right corner to nominate someone, including yourself potentially, if that's what you would like. Today's speaker is Tim Rieseberg. He was awarded his MS in chemistry in Göttingen, and he is just about to be awarded his PhD degree from that same institution, I think in four weeks, he told me. He has interests in chemical evolution and plant diversity, and you can see a few of the titles from projects that he has led on the right-hand side here. And I'll hand control over to him and let him take it away.
Yeah, thanks, Luke, for this kind introduction, and thanks to all of you for being here today. I'm really happy to present you some of the research of the past 4 years that I've did during my PhD. And today my title will be Carophyta, because today I will focus on carotenoids and their oxidative breakdown products, the apocarotenoids, and the role they might played in the Streptophyta, so streptophyte algae and land plants, for the conquest of land. And if you go outside, I think you're usually always surrounded by a beautiful plethora of land plants, and somehow they share one last common ancestor with this green slime here. And the question now is, how did this all came to be? So plant terrestrialization occurred almost 500 million years ago. And what you can see here is a simplified form of the family tree of the green lineage, including the chlorophytes and the streptophytes. And the streptophytes are comprised of the streptophyte algae and all of the land plants. And what you also already can see here is that the Zygnematophycean algae share one last common ancestor with all of the land plants. And with the projects I'm working on, I was part of a big consortium called MADLAND, where a lot of scientists work on different aspects of plant terrestrialization. And in my research, I mainly worked with 5 organisms, with the model moths Physcometrium patens, the 2 Zygnematophycean algae Zygnema circumcarinatum and Mesothelium antlicarianum, and the 2 Carophycean algae Cara tomentosa and Cara baltica. But for simplicity, I will from now on shorten them and call them P. patens, Zygnema, Mesothelium, and so on. And what basically happens is when you take the step from water to land is that typical terrestrial stresses like temperature fluctuations, light fluctuations, but also in water availability strongly change in amplitude and in frequency usually when you go from an aquatic to a terrestrial habitat. So since these terrestrial stresses usually strongly impact plastid physiology, land plants have developed a chemodiverse response to them. And with my projects, I was also working on main questions of our consortium, the features enabling the conquest of land, the molecular adaptations in early land plant evolution, and the molecular evolutionary drivers of tolerance to abiotic stresses. And more specifically, I was interested in the role of the carotenoids and their oxidative breakdown products, the apocarotenoids, in the conquest of land. And I want to start with a quick reminder why carotenoids are important and key players in plastid biology and where you can find them. So they hyperaccumulate in a lot of valuable crops, and they make me pretty happy when being extracted. And for example, in carrots, you have beta-carotene to a high extent, or in tomatoes, you find lycopene to a high extent. And these carotenoids are basically found in any photosynthetic eukaryote, and they are valuable to us because they are the primary source of provitamin A. But Why are they key players in plastid biology? Because they are core components of the photosystem 2 core complex, where they serve as important antioxidants, and they are also important for the light harvesting complexes, where they stabilize the xanthophyll pigments, stabilize different forms of the light harvesting complex. So, for example, in the light harvesting state, we find more viola-xanthine, while in the energy dissipating state, we find more zeaxanthine. So, carotenoids are essential for the function of the PS2 core and the light harvesting complexes. But also apocarotenoids are important and can be key players in stress response. How are apocarotenoids formed? Apocarotenoids are formed when we cleave a carotenoid on the polyene backbone via oxidative cleavage, either enzymatically or non-enzymatically. The most important enzymes are the so-called carotenoid cleavage deoxygenases. So if we, for example, cut beta-carotene here at this bond, we result in a large fragment and a smaller volatile fragment called beta-ionone. We know that due to its molecular characteristics, it can reach different cell compartments by passive diffusion. And some studies in land plants have shown that it is able to reach the nucleus, and other volatile apocarotenoids are also, and somehow elicit a change in stress gene expression, which leads to stress acclimation. How all of this exactly works still needs some future research, but we're getting some first clues about the mechanisms. And since carotenoids are present in any photosynthetic eukaryote, we thought it might be quite likely that utilizing signals derived from these carotenoids could be an ancient trait. And additionally, when plants terrestrialized, they again rose the oxygen levels 500 million years ago quite drastically again. And since these apocarotenoids are also formed via non-enzymatic oxidative cleavage, and terrestrial stresses increase the oxidative stress when you take the step from water to land, we think that due to terrestrialization, the first land plants have faced more apocarotenogenesis, so we thought it's quite likely that utilizing the signals could be quite aidful for an oxidative stress sensor for the conquest of land. Which also leads me to the overarching hypothesis of all of my research, basically. Streptophyta share core apocarotenogenesis and carotenogenesis functioning in stress mitigation networks. The question now is, where did parts of this network first evolve? In which last common ancestor did key components first evolve? To test this, we first looked for a chemodiversity of carotenoids and apocarotenoids in all of the organisms that we see down here. So the carotenoid pathway starts from the MEP isoprenoid pathway via important isoprenoid building blocks, isopentenyl diphosphate and dimethylallyl diphosphate. And then via condensation, we end up with geranyl diphosphate, which is an important knot in many terpenoid pathways. For example, from here, also part of the chlorophyll pathways derived. And as you can imagine, in photosynthetic organisms, we detected chlorophylls, but also the plastoquinone pathway originates from here. But if we condense two geranyl diphosphates, we end up with the first carotenoid of the carotenoid pathway. From there on, many desaturation steps happen. But before we talk about the next carotenoids in the carotenoid pathway, I first want to show you some nice red antheridia of Carophycean algae. And in some cases, we also see red brownish thalli. So the antheridia are the male reproductive organs of Carophycean algae, and it's a conserved feature that they appear in this red color. And in some species, we even get like red brownish thalli during summer season. So what we did is we took a species that stays green in summer season, but has this red antheridia, and we took a species that is monoecious, is dioecious, sorry. Here you have the male plant, which accumulates a lot of antheridia in the top. So we cut off the top parts to end up with a fraction that is basically comprised of almost only antheridia and some swollen end cells attached to them that are also really red. And we had an antheridia-free section. And then we thought, okay, could this be carotenoids? So we investigated the carotenoid pool of these Carophycean algae. And what we found was that rather usually rare carotenoids in the green lineage accumulated in this red intermedia, namely delta-carotene and gamma-carotene, which are the monocyclized intermediates on the way to well-known fully cyclized beta-carotene and alpha-carotene, which was quite surprising to us. And this nice finding of these rather rare carotenoids that we found in this Carophycean algae also resulted in a nice little publication, Conserved Carotenoid Pigmentation in Reproductive Organs of Carophyceae, that was really recently published in Philosophical Transactions B. So if you want to know more about it, you can also check here. So, but how did we detect all of these carotenoids? We used the reverse phase C30 HPLC method combined with a UV-Vis detector. And despite the high chemical similarity, so you can see that they're only distinguished by the position of this one double bond, except of that, that's the same molecule basically. We were still able to nicely separate them. And what you can also see in this chromatogram, that they really hyperaccumulated. So those are the two carotenoids with only one cyclization happening. And here, for example, peak 7, that is chlorophyll B for a comparison. So we detected them by comparing them to literature data. For carotenoids, you have good literature data in different solvents of absorption spectra, and they did nicely fit the absorption spectra. And as you would expect, the absorption spectra were pretty similar, still with only this one bond shifted, but there was a small shift and they did fit really well. And we also thought, okay, if we have such a high extent of monocyclized carotenoids, we have to have also the oxidative breakdown product of the linear ends, because that's then the mostly occurring group in our carotenoid data set. And indeed, in general, in all organisms, we detected the small volatile apocarotenoid called 6-methylheptanone that originates when you cut here the linear end or here, but only in the Carophycean algae we found it to really high extent in our GCMS spectra, so clearly dominated our GCMS spectrum. That's like almost close to saturation, and it was about 10 milligrams of biomass that I used here. So yes, we thought, okay, how can this be that this rather rare carotenoid accumulates in this Carophycean algae in the Antaredia? So we looked for the lycopene cyclases and computed the phylogeny and found, okay, it basically resembles also the genome phylogeny that we know. So, and the homologs were good. So we thought, okay, nothing special about the lycopene cyclases here from this perspective. But then we looked for their expressional level and found that they were in all samples almost depleted. So there was enough to still detect them and put them in the phylogenetic tree, but expression levels were usually really, really low in all of our samples. So this also makes sense from a perspective of lycopene cyclases B. Especially lycopene cyclases B is a fast-acting enzyme that usually almost in a go closes both of the rings of lycopene when it reacts with it. But in this case, with this really down-regulated transcripts, we think that it's this way possible that you can also accumulate the intermediate if enough educt is there. And indeed, enough educt, there was another hint also, lycopene seems to be converted quite fast. Indeed, we found also that one of the enzymes that is important for delivering enough electron-accepting cofactors for the educt, that's a common motif that you also find in land plants that hyperaccumulate carotenoids in fruits, or for example in maize kernels, we found that plant terminal oxidases were really upregulated to provide enough plastoquinone for desaturation steps again and again. So, but when we now close the second ring and go to famous beta-carotene from beta-carotene bearing carotenoids, there's quite some important volatile apocarotenoids that originate from them, including beta-cyclositral, which gets more and more an accepted retrograde signal, at least in land plants, and also beta-ionone and its oxidation product, dihydroactinidiolide. And we were able to detect those apocarotenoids as well in the streptophyte algae as also in the moss Physcometrium patens. So to sum up, we found quite some conservation on a biochemical level for carotenoids and apocarotenoids in all of the organisms that we basically looked at, with some divergence for this rather rare carotenoids that only accumulated in Carophycean algae. But next, we wanted to test this network under different abiotic terrestrial stresses. So we took 3 of these organisms, the 2 zygnematophycean algae from the group cistertual land plants, Mesothenium and Zygnema, and the moss Physcometrium patens, and subjected them to stress experiments. We subjected them to highlight stress, followed by a recovery phase, to cold stress, to heat stress, and we also performed a control timeline. And at this time point, I want to thank Niels, Sina, Janine, and Tanya for all the help with the stress experiments. And during these experiments, we performed time course sampling. So we harvested samples for different purposes at a lot of time points that are represented by this rectangular lines here and by the dots. So we harvested samples for RNA seq analysis, for metabolite profiling of carotenoids and apocarotenoids. We also did some photophysiological measurements and at some time points also investigated the morphology of the organisms. But today, I will mainly focus on the RNA seq data and on the metabolite profiling data of carotenoids and apocarotenoids. So to sum this data set up, these are 3 organisms from more than 600 million years diverging groups, and it's a pretty comprehensive transcriptomics and metabolite profiling data set that we then had in hand after we did this nice time series stress experiments. And at this time point, I want to thank Armin for the help with the bioinformatics work, and I want to thank Conny, Sabine, and Malte for help with the analytical machines. And this also resulted in an already available preprint that is currently under review. So when you want to look in this quite comprehensive transcriptomics and metabolite profiling dataset, you can check here. And there's also a website attached that you find in the publication where you can look for your own gene of interest and see what it does in our streptophyte algae and in Physcometrium patens. So if we have a look at the global transcriptome profiles, we get pretty similar trends in all of our 3 organisms. So here the same color code holds true. Heat is red again, highlight is yellow, its recovery is green, blue is cold, I think you get it. So what we can see is that highlight and heat usually did allocate the strongest and fastest response to stress, and that it follows some temporal progression, and that was the general motive. So we got a clear separation of global gene expression profiles by temporal progression of stress. So our stresses do different things, and our stress experiments work as it seems so far. But now let's have a look at the actual pigment levels. So if we look at the chlorophylls and the carotenoid pools under stress exposure, already directly what jumps in the eye is that the overall pool size of the algae Mesothenium and the moss Physcometrium patens are way more comparable than the pool size of Zygnema caercum carinatum. But if we look now at the response due to oxidative stresses that we applied, basically the same motive holds true in all of the 3 organisms and also in all of the 3 stress conditions. What changes is the speed of the response depending on the organism and on the type of stressor that we apply. To take a quite extreme example, we take Mesothenium in highlight conditions. There you see at control conditions, we have a clearly viola-xanthine dominated xanthophyll pool, and when we apply highlight stress, the pool clearly shifts over to almost only zeaxanthine from the xanthophyll pigment. The rest of the pool there's also some change, but the major change happens within the xanthophyll pigments. So highlight stress, but also generally the other oxidative stressors, heat stress and cold stress, shift the xanthophyll cycle towards zeaxanthine to a more energy dissipating state. If we now have a look at the apocarotenoids, also general similar trends in most of the organisms hold true for most of the stressors, with some exceptions, for example, for cold stress, for the overall ratio of carotenoids to apocarotenoids for Mesothenium. But otherwise, we see similar tendencies induced by the abiotic stressors on the small volatile apocarotenoids that we investigated. And what you generally can say is that due to oxidative stress, you shift the ratio of carotenoids to volatile apocarotenoids towards the volatile apocarotenoids. So we have declining values here. And what we also see is that we have the strongest response in Mesothelium generally and the smallest response we find in Zygnema. That is a general motive that we saw in stress experiments from different studies, that this is quite a tough organism that does not react that much to a lot of stressors. So, but what we also found is that highlight induces the strongest response in all of the organism on an apocarotenoid level, and that most of the change is happening conserved within the first 2 hours. So with all of these time-resolved metabolite profiling data of carotenoids and apocarotenoids and all of the time-resolved transcriptome data that we generated, we wanted to all combine this to get some biological meaning out of it and to see if we find some new connections. So we used quite a state-of-the-art method called sliding window inference for network generation combined with random forest that I will from now on call swing RF for simplicity to get a predictory network. And what you also directly can see in our predictory network, LRR kinases were pretty central. So what we have here is a network that basically is connected of genes and metabolites connected by the strongest predictions of transcriptional change. So the direct connection between 2 dots, either metabolite and gene or gene and gene, means that one is the best predictor of a transcriptional change of the other one. So, and with this network, in this network, as I told you, we found this LRR kinases that were pretty central. And due to the method, we know from that already, okay, there seems also to be additionally quite some strong transcriptional change. Sorry, I need a sip of water. Transcriptional change within our stress experiments in all of the 3 organisms, which was already a bit surprising to us because usually when you think of a kinase, you think, okay, it's there and then there's a environmental cue and then it does something. So this strong transcriptional change we did not expect at first hand. But we thought, okay, let's have a deeper look at this LR kinase network that is central in our predictory network, and let's also have a general domain search for these LRR kinases. And in an independent analysis, we also found several of these LRR kinases in the Zygnematophycean algae, but also in the moss Physcometrium patens, with generally larger numbers in the land plant, which follows a general motive in kinase evolution. But in this conserved hub, we found several candidates that we would expect to be connected to kinases like phosphatases or CDPK. But what we also found, and which made me pretty happy, is that the small volatile apocarotenoid beta-ionone was one of the strongest predictors of LRR serine threonine kinase transcriptional change. With that, we found a new connection of apocarotenoids to LRR kinases that was not known before and that is conserved over 600 million years of evolution. But how this all exactly works, for that we need much more future research to say how the exact interaction functions. But we still want to try to sum this up in one biological model. So we have seen that abiotic stresses have a strong direct influence that is well conserved in all of our organisms that we looked at on carotenoid and apocarotenoid levels and their relative ratio to each other. What we also know is that these small volatile apocarotenoids can travel to the nucleus by passive diffusion, and also our experiments showed they are able to induce some change in stress gene expression, which then leads to some stress acclimation effect. And in this central kinase hub that I did not talk in detail about those, we also found several transcription factor candidates that could possibly play a role in this network, but for that we also need further research. But what came quite clear to us is that there seems to be a connection between the transcription of kinases and beta-ionone, and this will also have some influence on the actual protein level, but for that we should measure some more actual protein data. But that's a good point, I think so, for a new start of more research of apocarotenoid LRR kinase connection. And interestingly, especially these LRR kinases are well-known members of kinases that are able to interact with small molecules as effectors. I think the most prominent example of LRR kinases that interact with small molecules are steroid receptors. So I think it's really worth to have a deeper look into this in the future. So to sum this all up, we detected several conserved apocarotenoids and carotenoids in all of the streptophyte algae and the moss Physcometrium patents that we looked at, with some really special rare carotenoids that we only found hyperaccumulating in Carophycean algae. We did stress experiments to elucidate the role and response to abiotic stressors, and we found a strong direct conserved influence of these abiotic stresses on their relative level. And additionally, we found conserved beta-ionone as a strong predictor of LRR kinase gene expression and some potential transcription factors like mTORs and so forth that could be part of this network. So, and I hope this made you a little interested in plant terrestrialization and the context of specialized metabolism and how it aided plant terrestrialization. So if you want to read something more about it, I can recommend two reviews that sum up a lot of also other specialized metabolisms and what we currently know about them in streptophyte algae and land plants. And in the end, I want to acknowledge quickly a couple people. I want to acknowledge Jan for offering me this nice PhD and giving me the possibility to do all of my research. I want to thank Armin for all the help with the bioinformatics and a lot more people for the help and the collaboration. I want to thank Professor Feusner and the whole group of Professor Feusner for enabling me to do my metabolite measurements. I want to thank Anja Holzhausen for the really nice collaboration on the CARA project. So we basically together imagined this project. I want to thank the University of Göttingen for hosting me, and of course Madland and the DFG for funding my project. Now I want to thank you for listening, and I'm looking forward to the discussion.
Great. Thank you, Tim. That was a nice talk. If anybody has any questions, feel free to unmute yourself and ask, or raise a hand, or put it in the chat. I can start with the first one. I was wondering about, this is a very interesting method. It's predicting the connection between ionone and LRR kinases. I was wondering is there any metric on the accuracy of these predictions? How confident can we be that it is in fact a regulator, and any confidence values or thresholds?
So yes, there were quite some studies that compared the predictability of these different computational methods that give you predictory networks at the moment. And this method, actually, this swing RF, was the method that quite much surpassed all of the methods that came out in the past 20 years. The thing was why you don't see it much in plants is that it takes a lot of computational power, and nobody did it before with something that has a chloroplast. So before it was only done with yeasts and with bacteria, because otherwise you usually take too much time. And I can tell you it also took us several months to get the result, and we occupied a lot of computational power. So from this side, I can already say it's at least the most powerful computational predictable method for these kind of things. But I don't know if I believe this like 100%. I take this with a grain of salt by myself, but some of the authors of these methods say it gets into the range of DAPSeq from the predictability. So we really need some biochemical assays to prove that and to solve the actual biochemical interaction we have here. But these networks give you really good hints on what to look for with quite some accuracy.
Thank you. Go ahead. Sabah.
Very nice talk. And I have
Thanks.
one question about this connection to serine threonine kinases. With this expression is done on leaf tissue. In this case, is algal tissue, all of this.
So yeah, it was algae tissue and the protonema phase of Fuscometrium patens. And that were like the conserved motifs that we found in all of the three organisms. So that was conserved over the 600 million years.
Okay.
Generally, we also did know before that we have these LRR kinases already in the Nematophyceae, so in the whole algae tissue, but we did not know what they exactly do in them.
Do you know the level of transcript for the kinase in those cells, or is it all depends on from a RNA-seq data?
So this is all on RNA-seq data. I don't know if somebody already, within the meantime, measured actual protein data about these LRR kinases in the streptophyte algae. But for each exact kinase, you can, in theory, check on our website. I cannot tell you for each kinase. So they were one of the ones that showed some strong transcriptional change in general. But the individual kinases, you can check in theory on the website and look for them and can follow up the time series.
Wonderful. Thank you.
Really welcome.
Any other questions?
Yeah, I have one question. I was wondering if the carotenoids that you found in the Streptophyta algae and also in the moss, are those structures known from any other angiosperm species, or are they unique to these streptophytes? I might have missed that in your presentation.
So most of the carotenoids that we detected are quite common carotenoids in most of the green lineage, I can tell you. So the xanthocyclic pigments are the classical ones that are used in most of the organisms of the green lineage, and also, of course, things like beta-carotene and alpha-carotene are quite well distributed. But this delta-carotene and gamma-carotene are pretty rare. You can find it to some extent in tomatoes, but I think this is a different mechanism. There you just have that much lycopene that you also end up with detectable amounts of delta carotene and gamma carotene. That examples you find in several specific examples where somebody measured some amount of it. But where you usually find it is actually in green photosynthetic bacteria and in psychrotolerant yeasts. That's where you find usually this high extent of gamma carotene and delta carotene, and not in many other organisms. So within the green lineage, to my knowledge, that's the only organism that really accumulates them to such high extent.
Yeah, that's really interesting. Thank you.
Welcome.
Okay. There are no more questions. Then let's thank them for this nice talk and for answering all the questions. See you all in 2 weeks.
Yeah, thanks for the nice discussion, and thanks for giving me the possibility in general. It was really a pleasure. Really a pleasure. Thanks.
Congratulations once again on your thesis.
Thanks.
Thank you.
Thank you.
Okay. Then